Says GCC CD S21 slides, enzymes and energy, 41421.
No, I don't have it. Maybe a few days my computer will have problem. Maybe on that time you send it that one.
I posted it on Monday, and it's in Canvas, so it should be accessible to anyone who has access to Canvas. Okay, anything else? There's only a couple of things I want to do today. This is one of them, go over that worksheet. So why don't we do that?
Gesundheit.
So we have it in slide number 7.
Okay, thank
We
you.
have, yeah, biological systems are never at equilibrium until they are dead.
Thank you so much. But I don't have the slides. If I had it, I could can find it.
And it's not exactly in, you don't have to do the slideshow. You will see it in the note on slide 7.
Do you log in and out of Canvas every day, or do you just leave Canvas logged in?
I am logging in Canvas.
Okay, because I'm seeing it under modules for week 12. It is the only entry for week 12, module 11.
I don't know because I checked many times. I couldn't find it. Now my Canvas doesn't work in my phone. I don't know what's the problem.
Gotta love Canvas.
Technology, ain't it great?
Yeah, it's great when it works. Can you see the worksheet?
Yes.
Yes.
Okay.
Yes.
Try to make it a little bigger. The first part of this worksheet was pretty easy for most of you. Number 1, those statements about enzymes, they're all true. The second, which one is not true, almost everyone got this right. Enzymes are consumed by the reactions they mediate. That is not true. Enzyme is a protein. Number 4, what affects activities of enzymes? Temperature, ionic conditions, and pH. All of them are correct. And then number 5, some of you had trouble with that. What letters on the graph would indicate the activation energy with enzyme? And the answer for that is C, where the activation energy is reduced. So what is B?
B is the energy required without an enzyme.
Without enzyme, right. So if you compare B and C, that's the answer. C is much less energy required because the enzyme is there. Now 6, cellular structures for converting carbohydrates into usable energy are mitochondria. Most everyone got that right. Number 7, energy for cellular respiration is ATP. Breakdown of glucose is done in mitochondria. And then this is the one we've been talking about. According to the second law, an organism whose metabolic systems are at equilibrium means it's getting no energy input, and therefore it's dead. Mm-hmm. And the last 3, there were a considerable number of problems that various people had. Number 10, two types of chemical reaction. Those require energy are called endergonic. Ender means going in, energy going in. Those that release energy are called exergonic. And then number 11, enzyme partners. Most of you got 3 of them, cofactors, coenzymes, and prosthetic groups. But also included in that is in subcategories are iron and vitamins. So on that list, there are really 5 that answer the question, enzyme partners. And the last one, anabolic pathways require input of energy to convert small molecules or simple molecules into polymers or complex molecules. And catabolic pathways are the reverse, the release of energy as they break down large or complex molecules into small or simple ones. And quite a number of you had that reversed, that you had the anabolic pathway converting large molecules to small, and catabolic the opposite. So make note of that, which is which. Any questions about this worksheet beyond what we've already discussed? All right, then we will stop sharing that. And you should see overview of where we're going. So we're back to this same introductory slide because I want to continue to set the context for you. We're talking about 4 main topics: energy, enzymes, photosynthesis, and respiration. We've already talked about energy. What is it, where it comes from, what it's used for, and enzymes, how they work and what regulates them. Today, the emphasis is going to be on photosynthesis. Now, there'll be a little bit of respiration worked into this because photosynthesis and respiration are tightly associated, even though they are opposites of each other. So photosynthesis is the capture of energy from sunlight and conversion of it into chemicals. Respiration is the reverse of that. It's the derivation of energy from food that's been created by the force of sunlight. Photosynthesis. All life on Earth requires a constant input of energy. Remember the second law. That source is, for the most part, the sun. Green plants are the producers. Animals are consumers only. Animals do not produce. Their food source is either plants or they can eat each other. And if they're eating another animal, in all likelihood, the animal they eat ate plants. So food chains always start with plants. Just want to make you aware that on this planet, there is another source of energy capture besides the sun, and that's called chemosynthesis. So in contrast to photosynthesis, chemosynthesis is a form of hydrothermal energy. It's similar to photosynthesis in that the starting products are the same, carbon dioxide and water, in both cases. In the case of photosynthesis, the energy input comes from the sun. In the case of chemosynthesis, it comes from hydrogen sulfide. I have some pictures here of organisms that live in thermal vents that are so deep in the ocean that sunlight never gets there, so they have to have another source of energy. And that, in most cases, turns out to be hydrogen sulfide. And the end product, instead of being oxygen, is sulfuric acid. So this is just a side comment. I just wanted to make you aware of the fact that not all energy capture comes from the sun, but most of it does. Okay, we'll start with this overview of photosynthesis. Can divide the processes in photosynthesis to two categories called light reactions and dark reactions. The light reactions are the ones that result from the capture of sunlight. Sunlight is received by pigments in the chloroplast and the thylakoid membranes. That results in the creation of ATP and NADPH, which go to another part in the chloroplast called a dark reaction, where carbon dioxide is fixed and makes sugar. The light reactions generate electrons, which are then transferred via the electron carrier NADP to the dark reactions, which occur in the Kelvin cycle. So I want to tell you in more detail next about the details of the light reactions and the details of the dark ones. The chloroplast structure. Well, here's a plant cell called a mesophyll cell. And you can see familiar structures, the cell wall, the nucleus, this large vacuole. And then these little green things are the chloroplasts. Within the chloroplast, you can see these stacks of structures. These are called grana. The entire stack is called a grana. And within the grana, it's made up of these stacks of membrane-bound vesicles known as thylakoids. And it's within the thylakoids that the light reactions occur. That's where light is received and energy is generated. The dark reaction, on the other hand, occurs in the stroma. The stroma is the space between the thylakoids. So the thylakoids provide energy so that the Kelvin cycle, the dark reactions in the stroma, can occur. And it's the thylakoid membrane. Remember, we end up with chemiosmosis. It's the thylakoid membrane where the electron transport chain is. I just mentioned that the dark reaction involves the Kelvin cycle. And this represents that. If you start at the top, the input is 3 molecules of carbon dioxide. And there's a process known as carbon fixation. Carbon fixation is a process by which plants convert atmospheric carbon from the gas carbon dioxide to form solid organic compounds, such as glucose. So that's, when you think about it, that's a pretty amazing thing, that the chloroplast can take a gas and make a solid out of it. carbon from carbon dioxide and make glucose. The critical first step is an enzyme called rubisco that catalyzes the first step of carbon fixation, whereas the atmospheric carbon dioxide is converted to energy-rich molecules such as glucose. And this occurs in a cycle. So three molecules of carbon dioxide go into a 3-carbon compound. Then it requires ATP, energy input, to convert that to the next intermediate. It requires energy from NADPH to do the next step. And finally, you get to an intermediate called glyceraldehyde 3-phosphate. So this is a 3-carbon molecule. And that then is released from the Calvin cycle, so that the stoichiometry here is that if you start with 3 carbon dioxides, there's 3 carbons here, you get 1 glyceraldehyde 3-phosphate molecule out of that. And so each of these 3 carbons, I've got the structure here of glyceraldehyde 3-phosphate. Each of these 3 carbons is from the one of the 3 carbons in carbon dioxide. So that one turn of this cycle costs 9 ATPs plus 6 electrons from NADPH. So I hope you get the picture here that that we have, on the one hand, these light reactions where we're generating energy. That energy goes into the dark reactions where sugar is made. Well, photosynthesis and respiration are coupled in a sense, and that one is the reverse of the other. Photosynthesis is really glucose synthesis. It's a metabolic pathway that's primarily anabolic. It requires energy to go in, where carbon dioxide and water are used to make glucose and oxygen. Respiration is glucose oxidation. It's a metabolic pathway that is primarily catabolic. That is, energy is released when glucose is oxidized to produce carbon dioxide and water, which are the starting points for photosynthesis. I'm going to re-emphasize redox reactions. So this is a review of information that we've gone over before when we talked about energy. Energy is harvested through electrons that are moved from one molecule to the next. These electrons can carry energy, some of which will be transferred to intermediate molecules as the electrons travel on a so-called downhill journey. The basis for these transfers is called redox reactions, oxidation and reduction. Some substances more strongly attract electrons than others. A substance that loses one or more electrons is said to be oxidized. A substance that gains electrons is said to be reduced. These two processes always occur in pairs. If there's an oxidation, there's always a corresponding reduction. You've seen this before, but it's a simplified version of redox. If you have a substance A that is reduced, it has an electron, which it can transfer to substance B, which is oxidized. And when that happens, that is now reduced. So after that happens, A is oxidized and B is reduced. And if we look at this in a biological sense, we can take an organic molecule, for example, glucose, which is reduced. It has two extra hydrogen atoms. These can be transferred to an enzyme, an electron carrier, the coenzyme NAD, or in the case of photosynthesis, NADP, so that you end up with an oxidized molecule, organic molecule, and reduced carrier, plus an electron. Now, Scott found a video I'd like to try to show you.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the four complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits two high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal two reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality, they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
Are we back with the slides?
Yes.
Okay. That video was got into some detail that is beyond what we want to go into, but the point of it is to show the electron transport chain within a membrane. And it doesn't matter whether that membrane is in the chloroplast, in the thylakoid, or in the mitochondrion. It's the same type of process in both. It's how electrons are transferred from one carrier to the next, so that, let's see. This is what's happening. This is a simplified diagram, but if you have a substance that can donate electrons and a carrier, the carrier can release energy and also transfer electrons to the next component of the pathway. So in those protein complexes that were shown in the video, this toggling back and forth between one electron carrier and the next, that's what's going on in within the protein complexes in the membrane. Okay, ways to produce ATP. There are three: substrate level phosphorylation, which is direct enzymatic transfer of a phosphate to ADP to make ATP. Oxidative phosphorylation, which is the transfer of electrons from organic compounds like glucose through an electron transport chain to inorganic molecules like oxygen. Chemiosmosis generates the ATP as the energy is released in oxidative phosphorylation. Photophosphorylation, pigments of photosynthetic cells trap light energy and convert it to chemical energy, again through an electron transport chain and chemiosmosis. So the difference between oxidative phosphorylation and photophosphorylation is really how they start. This is, again, you've seen this before. This is substrate level phosphorylation. It's the first type, first method by which ATP can be made. You have one molecule of phosphoenolpyruvate plus one ADP, and you get one pyruvate molecule plus one ATP. So what is pyruvate? It's glucose split into two. When the glucose molecule is split in two, it becomes two molecules of pyruvate. Now, the second way to produce ATP is photophosphorylation. It produces glucose in the chloroplast. So pigments in the photosynthetic cells trap light energy, convert it to chemical energy through an electron transport chain, plus chemiosmosis, plus carbon dioxide fixation. And this illustrates light coming in, chlorophyll absorbing it, which splits water into two protons plus 1/2 an oxygen molecule. This releases excited electrons, which then go into this electron transport chain, which you can visualize as a downhill series of steps. And each step is a redox reaction. So each step, if we can go back, go the other way. Here, each step down those stairs is one of these. And finally, and so it's during this downward stepwise process that ATP is generated through chemiosmosis. And the final electron acceptor is NADP to become NADPH, and that goes to the Calvin cycle, which we'll talk about, which makes glucose. Scott, is there anything you'd like to add to anything I'm saying here?
No, sir. It all makes sense.
Okay. Well, okay, the final way to produce ATP is oxidative phosphorylation. There's energy from glucose via respiration in the mitochondria. Electrons from organic compounds like glucose are passed through a series of reactions, again, an electron transport chain, to an inorganic molecule like oxygen. And involved at the end of that is chemiosmosis, which is the mechanism that actually generates the ATP. And just to remind you, you've seen this diagram before, illustrating very simply what chemiosmosis is, where you have potential energy, a high concentration of ions on one side of a membrane, low concentration on the other. ADP is converted in this membrane by an enzyme called ATP synthase into ATP. So let's go through oxidative phosphorylation. It starts with glucose. The process of glycolysis of glucose breaks glucose into two molecules called pyruvic acid, pyruvate, and these can be converted to acetyl-CoA, and this is the start of the Krebs cycle. And I'll show you the Krebs cycle in a bit. But at the level of glycolysis, there's a substrate level production of ATP. So you get two ATPs at this step. Then, as the process goes on, NADHs are generated at various different steps, and they're accumulating on one side of a membrane until finally the electron transport chain can start, which results and ends up with chemiosmosis, and finally the electrons are transferred to oxygen, and that makes water. So you get more substrate level ATP synthesis from the Krebs cycle, but from the electron transport chain and chemiosmosis, you get a lot. In this case, 34. So starting with one molecule of glucose, you get 2 ATP substrate level ATPs, 2 more here, 34 here. So a final total of 38 ATPs from one molecule of glucose. Is that clear what oxidative phosphorylation involves? And again, Scott, if you have something to add, go ahead.
No, just in general, it's important to start to visualize where all these different things are happening, in what structures, in what kinds of cells, where in these structures are things happening. Because if you can start to form this picture in your head, it becomes a lot less abstract. And I find that usually helps people. So really try and pay attention to where things are happening. And that kind of explains why things happen the way they do.
Okay, I have some pictures coming up, I think, that will illustrate what Scott's saying. But first, here's a table contrasting oxidative phosphorylation and photophosphorylation. So oxidative phosphorylation is the process that produces ATP using enzymes and oxygen. Photophosphorylation is the process of ATP production using sunlight. What's the source of energy? For oxidative, source of energy is glucose along with molecular oxygen. In case of photophosphorylation, source of energy is sunlight. Location, oxidative is in the mitochondria. Photophosphorylation is in the chloroplast, which as Scott has just mentioned. It's important that you keep these cellular locations in mind. And I'll show you a couple of pictures in a little bit. The occurrence, oxidative phosphorylation occurs during respiration, whereas photophosphorylation occurs during photosynthesis. Even though this discussion is focused on photosynthesis, you can see that it's so closely coupled with respiration that you really can't consider one without the other. The final electron acceptor in the case of oxidative phosphorylation is oxygen, whereas with photophosphorylation, it's NADP, which is converted to NADPH. I've summarized here at the bottom, photophosphorylation starts with light and water. The light splits the water molecule to generate electrons, which along with carbon dioxide ends up generating glucose and oxygen. Oxidative phosphorylation starts with glucose and oxygen. It releases energy, ends up with carbon dioxide and water.
Excuse me, teacher, I have a question here.
Uh-huh.
Oxidative phosphorylation, does it occurs in plants or only in human and animals?
Oxidative phosphorylation?
Yes.
It's a good question because many people don't realize, they think, okay, photophosphorylation occurs in plants, but it turns out that it also, oxidative phosphorylation also occurs in plants. So plants have both photophosphorylation and oxidative phosphorylation. Plants have mitochondria, whereas animals have only oxidative phosphorylation.
Thank you, teacher.
Anything else?
I've thought of a maybe decent way to think about it. So it would be like you taking, like getting a drink of water from the tap using a cup, with the water being the energy. The energy is stored in the cup, but then you still have to drink the energy from the cup to get the water.
Uh-huh. Yeah. Okay.
That's one way I thought of it.
Good. Thank you, Zach. All right, let's move on. Now we're talking about respiration, and so you need to know something about, a little bit about respiration now, and then we'll talk a lot more about it next time. But the respiration is the main part of it is this cycle called the citric acid cycle, or the Krebs cycle, or the TCA cycle. All those names refer to the same thing. Well, it starts with this molecule, actually it starts with pyruvate, a 3-carbon molecule, which was made from glucose. So 2 molecules of pyruvate for 1 molecule of glucose, that's converted to acetyl-CoA, which then enters the Krebs cycle. And what the Krebs cycle does is release carbon dioxide and generate NADH plus electrons, which then there can be a substrate level phosphorylation as part of the Krebs cycle. But the main thing it does is generate electrons for electron transport chain. The cycle itself only produces a small amount of ATP. So the function of the Krebs cycle is to oxidize glucose derivatives, fatty acids, and amino acids to carbon dioxide through a series of steps. Those steps are in the cycle. And the result is high energy electrons from these oxidized fuels are transported by activated carriers, the electron transport carriers, NADH and FADH2, to the electron transport chain. And that's where we talked about before, that's where most of the energy is generated. So the Krebs cycle generates a little bit of ATP. But the most important thing it does is produces all these electrons, which can go into the electron transport chain. And there, along with chemiosmosis, the bulk of the ATP molecules are made. So from one molecule of glucose, you get 4 from substrate level and 34 from the electron transport chain from oxidative phosphorylation. Now, Scott was talking about making sure you know where in the cell these things are happening. Well, this is oxidative phosphorylation, citric acid cycle. It's called the TCA cycle or the Krebs cycle or the citric acid cycle. This occurs in the mitochondrial complex. So this picture is a drawing of a mitochondrion within which are these membranes. There's the membranes are called cristae. And the inner mitochondrial membrane is where the electron transport occurs. And it's in the matrix. Well, the important point is that electron transport chain and chemiosmosis occur in this inner mitochondrial membrane. So that's oxidative phosphorylation. Let's see. I'm going to skip this for now. Ways to produce ATP. This is a review. We've gone over that already. But I want to show you this comparison between the Krebs cycle and the Calvin cycle. Calvin cycle occurs in chloroplasts, the Krebs cycle in mitochondria. And these have to be considered together because they're interrelated. So the definition, the Krebs cycle is part of aerobic respiration. The Calvin cycle is part of photosynthesis. It's the dark reaction of photosynthesis. Location, the Krebs cycle occurs in mitochondria, Calvin cycle in chloroplasts. The Krebs cycle is catabolic. It tears things apart and releases energy. Calvin cycle is anabolic. It uses energy to build up molecules. ATP and carbon dioxide are produced in the Krebs cycle. In the Calvin cycle, they're used. Now, the next point is relevant to the question you asked. The Krebs cycle takes place in all aerobic organisms. So that means that both The Calvin cycle or photosynthesis occurs only in plants, but plants also have respiration, whereas animals only have respiration. So animals have only the Krebs cycle. Plants have both a Krebs cycle and a Calvin cycle. The Krebs cycle oxidizes glucose. The Calvin cycle makes it. And finally, the Krebs cycle respiration occurs in the presence of oxygen, whereas oxygen is not involved in the Calvin cycle. So these these comparisons are things that you should study and make sure you understand them. Okay, we're going to come back and look more closely at the electromagnetic spectrum. How can we capture and make use of energy that is available in the electromagnetic spectrum? Well, here it is. You've seen this before. Invisible light is this very small part of the electromagnetic spectrum, and we see it as different colors: red, yellow, green, and blue. Now, for the purposes of photosynthesis, the pigments in plants can absorb red, yellow, and blue wavelengths. They cannot absorb green. Green wavelengths are reflected, so that to us, most plants look green. And this is a detail of why. Here's an absorbance spectrum plotted against wavelength. And you can see that if you look at chlorophyll A and chlorophyll B, chlorophyll A is the blue line. Chlorophyll A absorbs at about 680 nanometers, and again at a longer wavelength. Chlorophyll B follows the same pattern, but it has slightly different absorption optima. But you can see that neither of them absorbs in this green area. So that's why green light is reflected. Well, this is the structure of chlorophyll A. It's a pigment. Plants make a lot of pigments. In addition to the chlorophylls, xanthophylls, and carotenoids, and anthocyanins. And I've got a picture here of anthocyanins because these other pigments can often mask the presence of chlorophyll. So last time, you remember I showed you a picture of a red maple, which looked entirely red. And it's not because it didn't have chlorophyll, it's because the chlorophyll was covered up, it was masked by anthocyanins, which can vary in color depending on the pH. They can be red or purple or blue. So again, coming back to where in a plant does photosynthesis occur? It occurs in a leaf. If you look at a cross-section of a leaf, you can see various different kinds of cells: epidermis, stomates, and mesophyll cells. And this is where most of the photosynthesis occurs, in these mesophyll cells. So here is a drawing of just one cell. You can see has chloroplasts, and showed you this before, but to remind you and re-emphasize the point, within the chloroplast, there are these grana. You remember grana are stacks of thylakoid membranes. And within a thylakoid, there is the intercellular compartment, interthylakoid compartment, and the thylakoid itself is both the compartment plus the membrane. The light reaction of photosynthesis goes on in the thylakoids. The dark reaction of photosynthesis goes on in the stroma. The stroma is the space between the thylakoids. Thylakoids are stacks of membranes. So a chloroplast is really a combination of stacks of membranes and then space between them called stroma, where the Calvin cycle occurs. This is where glucose is made. Carbon is fixed. Okay, anybody have any questions about any of this? Or Scott, anything you want to add?
No, sir. I think at this point it's just practice, going over it, reviewing it, draw pictures.
Okay.
I have a quick question.
Yeah.
To what level of detail do we need to know Calvin and Krebs cycle?
What level of detail?
Like, do we need to know each step along the way, or do we need to just sort of understand the general idea that, hey, yeah, this produces ATP and carbon, or uses it, or
Well, you should be able to associate, for example, from this table, associate the various different processes with which cycle.
okay, so we don't need to know the individual steps within each cycle, just what each cycle does.
Right.
Okay, cool.
The stages in photosynthesis, you have these light reactions and dark reactions. So you've seen this diagram on the left before. The Kelvin cycle is expanded, elaborated a little bit on the right, where carbon dioxide goes in, and there's this enzyme Rubisco, which can take carbon from a gas and make it into a solid sugar molecule. So out of the Calvin cycle comes sugar. Now, this is detail that I don't expect you to know. I'm showing you this to emphasize that there is a lot of detail in photosynthesis that is going on that we don't need to know about at this level. But within the thylakoid, there are two photosystems, photosystem 1 and photosystem 2, and they're based on two different forms of chlorophyll. They absorb at different wavelengths of light. So light, a photon of light enters a thylakoid. In photosystem 2, excites electrons which go into an electron transport chain where energy is made, ATP is made, and electrons come out of that at a much lower level of energy and enter photosystem 2, which is a different form of chlorophyll, which absorbs at 700, and more light excites the electrons again to a high level of energy. And here they go into the Calvin cycle, where carbon is fixed and sugar is made. You don't need to know about cyclic electron flow. And this, I wanted to emphasize what's going on in the thylakoid. So if we go back to these thylakoids, And if you look at, let's say, just one of these thylakoid membranes like this, here it is. So here's the thylakoid, and embedded in it are these large protein complexes, which include photosynthetic pigments, photosystem 1 and photosystem 2. Light from the sun comes in, excites electrons. They go through an electron transport chain within the membrane. Chemiosmosis occurs within the membrane, and ATP is generated. In this case, what we're getting out is electrons that go to the Kelvin cycle to support the synthesis of glucose, of sugar. So this is the thylakoid compartment. This is the membrane. Outside the membrane is the stroma, and that's where the dark reactions occur, the Kelvin cycle. And this point here, it's making the same point again, that electron transport and chemiosmosis occur within the membrane. And you have this accumulation of electrons or protons on one side of the membrane. And when they go through by chemiosmosis, ATP is generated. Overview of light reactions. Oxygen is produced as a byproduct of splitting water. Solar energy is converted to chemical energy in the form of ATP and NADPH. The ATP and NADPH are used to power the light-independent reactions, that means the dark reactions, the Kelvin cycle, in photosynthesis. That's where glucose is made. This restates it. The light-independent reactions are known as the Kelvin cycle. Fixes carbon by taking carbon from carbon dioxide, a gas, converts it into solid sugars. And glucose then can be built up into polysaccharides, for example, starch, for storage in the plant. So when we eat plants, much of what we're getting are the polymers, the polysaccharides made from glucose that is made in the Kelvin cycle. Just an overview, simplified version of the Kelvin cycle does. It takes in carbon dioxide, spits out sugar, requires input of ATP and NADPH from two different sources in order to make sugar. And this is the Kelvin cycle overview. You don't need to know all these intermediates. The main thing you need to know is that carbon dioxide goes in. Rubisco is the enzyme that fixes carbon, and out comes glyceraldehyde 3-phosphate, which is then converted into glucose. And you've seen this before. And that is the end of the slides. Any question about anything that we've gone over today? The emphasis here has been on photosynthesis, but respiration was also included. The next time we'll focus on respiration almost entirely. Okay, our time is up. Remember that on Monday is the test. The Monday test, test 3, will not include photosynthesis. It will include cell division, meiosis and mitosis, Mendelian genetics, and energy and enzymes. But it stops with energy and enzymes. It will not include photosynthesis.
So cell division up through enzymes.
Yeah,
Okay, cool.
Wait, Professor, you said it was what date for the test?
that's Monday.
Do we have class that day?
Is Monday a holiday?
No, but I'm asking since we have a test. Sorry.
Oh, I see what you're saying. No, class will not meet on Monday because you have all day Monday to take the test. The test will be a full class period, but the class will not meet.
Okay, thank you.
Thank you.
Is there a study guide for the test, Professor?
Pardon?
Did you mention anything about a study guide, or is everything you listed just brief? Just like just go over the slides on the stuff you just talked about right now.
Right. The slides and go over the recording and the worksheet.
And the worksheet. All right, perfect. Thank you so much.
Yeah, thank you for reminding me. I'll send an announcement telling people not to show up for class on Monday. Okay, anything else? And I'm going to stop sharing. And I'll see you a week from today. I won't see you on Monday because you'll be taking the test.
Thank you, teacher. Have a great
Sounds
day.
good.
Thank you, Professor.
Okay, take care. Bye.
Have a safe weekend.
Okay.